To start off, the signal goes through an axon, and to pass the axon cleft, there are many small vesicles containing a neurotransmitter substance called acetylcholine. And when the impulse reaches the end of the axon, the neurotransmitter is released. It diffuses into the muscle cell membrane to combine with the receptors there. The binding of the neurotransmitters with the muscle membrane receptors causes the membrane to become permeable to sodium, resulting in the influx of sodium and the contraction of the muscle cells occur. When the muscles relax, the binding sites on the actin are covered by tropomyosin, and when the membrane of the muscle is depolarized, calcium ions are released from a system of tubes in the muscle fibers. The calcium ions bind with the troponin, and this displaces the tropomyosin from the actin binding site, as you can see now. The myosin head binds to the actin using the energy from ATP, and it forms an actomyosin bridge. As the actomyosin heads attach to the actin filaments, they tilt, causing the actin filaments to slide past it. As the actin filaments move past the myosin's heads, the sites become detached and are ready to attach to the next binding site. Then calcium ions are absorbed back into the tubes, and the troponin reverts to its original state. Tropomyosin again blocks the binding site on the actin filaments. During muscle contraction, the myosin head groups attach to the actin and pull it towards the center of the sarcomere. This causes the sarcomere to shorten. This just shows the general structure of a sarcomere. You have the myosin, which are indicated by the purple strings, and on the ends how it's crimped, it represents the myosin heads. You have the actin, which is the yellow that the myosin attaches to, and then wrapped around that on each end is the Z disc.